we're going to move on to our second talk, and I'm going to turn it over to Jan to introduce our second speaker.
Yes, thank you, Janine. I now have the task to introduce Richard Henderson. He will talk about the LMB, its unique history, and what might have caused some of the outstanding successes that have been produced here. I don't really have the time to introduce Richard properly, so I'll just give you a few key facts. Richard started at the LMB as a PhD student in 1966, so that's more than 50 years ago, and he's been here since then. He started using crystallography, but then quickly, encouraged by Nigel Unwin, switched to electron microscopy, which he has been doing since then. And together with Nigel, he solved the first structure of a protein by electron microscopy, bacteriorhodopsin. But that was using two-dimensional crystals, which is sort of quite a special situation. But in 1995, he published a very influential review that demonstrated from first principles that it should be possible to solve atomic structures by EM by not using crystals at all, which is single particle averaging. Since around that time, he and people around him have systematically worked on the many issues that might have caused EM to remain globology, despite the promise as published in his review. Almost as a distraction, I hope there's some insight, Richard became LMB director in 1996, which he did for 10 years. And there were many achievements, but the most important achievement, I think, was that he organized the LMB's new and expensive shiny building that we inhabit now. And after that, he went back to work and was trying to make a single particle EM into a mainstream method. And as you all know, that worked out, and it turned out that really developing direct electron detectors, which he was quite heavily involved in, was probably the main reason why that happened in the end. Richard was at the heart of an amazing few years at LMB and elsewhere. That meant that single particle EM went from 4 angstroms then to 3 angstroms and is now below 2 angstroms. Richard is still active in the lab after more than 50 years. He's a fellow of the Royal Society, member of the National Academy of the US, and in 2017, together with Joachim Frank and Jacques Dubochet, won the Nobel Prize in Chemistry. And I hope I haven't used up too much time. Richard, they're all yours.
Hopefully I'm now live and in action. So it's a great pleasure to join this great new intercontinental seminar series that's been led by Janelia Farm. I think I'd like to tell you a bit today about the Medical Research Council Laboratory of Molecular Biology, or LMB as we call it for short. As Jan mentioned, I've been here since being a student in 1966, so over 50 years. So it's a little bit difficult to fit all of that time and all the different events into a 15-minute talk. So what I've decided to do is to pick 3 short periods and give you a little snapshot of how things were when the LMB started, and then how it was and how things had changed during the 10 years that I was the head of the lab. And then in the third part, sort of try and come up to date and perhaps mention 1 or 2 of the issues that are very important in the continuing evolution of the LMB culture. So I'll begin just with the first slide. Is this is a timeline of the Mrc Lab from the time 1947 when Max Burutz and John Kendrew got their very first grant from the Mrc for 5 or 6 people, right up to the present day. And it has in it pictures, I won't mention them, but hemoglobin and myoglobin and the original DNA double helix model of Watson and Crick, right up to the more modern time with monoclonal antibodies, the ribosome structure, and then leading into the work that Shores and Michel have talked to you about today. But the person that was probably most important in getting the lab off to a flying start and then maintaining its focus on being successful in research was Max Burutz. So he came from Austria as a PhD student in 1936, set himself the goal of working out the structure of hemoglobin in the blood. And it took him till he was successful. 1968 took 30 years. But during that time, as well as being a successful scientist, he learned to appreciate the suggestions, the insights, and the power of other people that he was interacting with. So when the time came to move from their very restricted quarters in the physics department in Cambridge to create a new lab in 1962, Max was keen to have a place where people could interact. And this is Max in the canteen. Max's wife, Gisela, was the manager of the canteen for 10, 15 years. And here he is chatting to some cell biologists, genomics people, as well as structural biologists. And Max is standing because he had a bad back, but everyone else is sitting and they're all perfectly happy. And so over the years, Max was asked what was his philosophy and what was the culture he was trying to inculcate into the lab. And he had an opportunity to write a number of articles. This was from an article in the medical journal. His view was the key thing for both the head of the lab and the people in it was to have a deep and genuine interest in the science. Science was the focus. And then to choose important and interesting problems rather than derivative ones. Find the best people you can to teach you, your best mentors. And then if you later on get into a position of responsibility, recruit people who are preferably even cleverer than yourself, be interested in everybody's work. And then two key things was, one, make sure you are well funded. So if your experiments run into trouble, you don't want to run out of money halfway through before you can fix the experiments. And fortunately, the Medical Research Council in those early days kept funding the lab, even though it was extremely difficult to get solutions to the problems that they were trying to address in the early days. So funding is very important. And then later on, as part of the effort to have seminars like this one, we thought it'd be good to have a lecture theatre in the lab and got the money for it. And we asked Max, would he be happy if we named the lecture theatre the Max Burutz Lecture Theatre? And he agreed, provided that we put his motto on the wall. And we said, oh, Max, we didn't realize you had a motto. And there it is. His motto is, in science, truth always wins. And he explains this in this video.
And one of the great things about science is that truth always wins. And that makes it different from many other subjects where about history and economics and philosophy. People can argue for generations. And this is one of the things which is least understood by the public and the media, because the media always think that, as say in politics, they have to bring together two opposing views. But in science, there are not two opposing views. Nature always tells you what's right, and that is final.
So that gives you a kind of impression of Max's warmth and his support for other people's work. But other people were there at the lab at the beginning. And one of the others, I thought I'd tell you a little bit about France. What did Francis Crick think was important in a lab in terms of the culture? And of course, they were in very restricted quarters in the early days. So he characterized it as ruthless research in a cupboard. But they then persuaded the Medical Research Council to fund this new building. And the concept of the new building was to bring people with wider disciplines together. So for example, Fred Sanger and his colleagues from the biochemistry department in Cambridge were then being merged with Burutz, Kendrew, Crick, and so on from the physics department. They also recruited people from outside, from London. So Aaron Klug and Hugh Huxley came from London. So it was a merger of people from different disciplines with the goal of bringing them together and extracting more value out of that collaboration than you get from individually. But a lot of the success depended on getting a lot of talented and capable young people. And in the beginning of the lab, more than half of them were postdocs and students who came from the USA. In fact, in those early years, 1962, 1970, there were more students and postdocs working in the lab doing the research from the USA than from any other part, from the whole of the world put together. And in fact, more from Harvard than there were from the rest of the UK. So that was very important. And then those people often came for a year or two, and then went off and set up their own labs and were quite successful. And then they were keen, particularly Crick, on having, for supporting Max with the canteen, encouraging people to talk. Crick himself went there every day. We learned to love his rather loud and garish laugh. But one of his other innovations was to introduce the idea of having a photograph of everybody in the lab posted, so that if you were talking to someone, you wanted to know who they were, you can go and find out who they were afterwards by looking at their photo. And then every year, to create interchange of information about what people are doing, we have a week of seminars, an annual seminar symposium, which has been nicknamed Crick Week. It was most of the week, still leaving time for experiments, and that's carried on from 1962 right up to the present day. In the beginning, everybody could speak because the lab wasn't so big. Now, perhaps only 10 or 15% people get a chance. The lab's got too big. And then his view was that you should tackle important and difficult problems, possibly conceived of as impossible, but by jumping in and tackling them head on, you rapidly find out which ones were truly impossible and which ones were only apparently impossible. And then rapidly you got a solution for that. And again, MRC, in addition to providing the funding, they didn't tie them to the projects that they said they were going to do. There was flexibility in the funding so that you could move to another topic if one wasn't working so well. And then one of his other bright ideas was that the funding agencies should be rewarded by having a prize. I don't think this has been introduced. For those who funded the most fast, that was to encourage them to be bold and rather than being conservative. So that was a little bit from the past. If we jump now to 1996 to 2006, when I was being the head of the lab, many things had changed in those years. So 1960 to like 35 years. So one of the big changes was that, built on the success of the DNA sequencing technology from Fred Sanger and others, sequencing viruses, bacteria, the nematode, in the year 2000, we had the human genome draft sequence, and then 2 or 3 years later, the full agreed sequence, and that created the scope for lots of new approaches that were not in the tradition of the early molecular biology years. So, and then alongside that, many of the discoveries that had been made and had been translated into patents or company startups started to produce significant amounts of commercial income through patents, royalties, and share sales. And these two things together helped the LMB to persuade the MRC that we should expand into transgenic mouse work. And there was a new mouse house facility with people to look after them, and then a new building which was opened 6 or 7 years ago on the back of commercial income from inventions that were not the prime goal in the early years but became so. And then also in 2003 there was a big bang in British science, whereas before that each scientist employed by a particular research council could only get money for their research from that research council. It was kind of demarcated. So Wellcome Trust had to apply for Wellcome Trust, medical research to medical, but there was a deregulation so that anyone could apply to any funding agency, and the success of the award would depend on the merit of the science. And so that opened up more flexibility in funding. And these were the main events that changed the approach in terms of the lab. And so one of the things was company startups, and here are a dozen or so companies that were started either by people in the lab or with input from people in the lab, and the ones that are highlighted produced significant sums of money, particularly Greg Winter's work on humanizing and on phage display. And that produced a lot of success, which proved that molecular biology, rather than being an esoteric academic pursuit, also had practical benefits into human health and sort of national enterprise. And so in 2003, which was the 50th anniversary of the DNA double helix discovery by Watson and Crick, there were lots of meetings, lots of celebrations to mark that 50th anniversary. And I was asked to write a little article for the Royal Society newsletter. And based on looking at the trends and how things had been changing, I said it looked to me like there was quite a systematic move from discovery in biology. So in the early LMB, everyone was discovering things, focused on, they might invent techniques, but it was focused on discovery biology. As time went on, first one group, then two, then three, and now we're probably up to 6 or 7 groups in the LMB are focused on invention rather than discovery. So new techniques, so synthetic biology, monoclonal antibodies would be 2 good examples. So there's a change, a lot more interaction between academic labs, whether it's a university or an institute, and companies, people going into companies, out of companies. And that was quite a big change in the culture, which of course helped us with funding and things like that. So jumping now to the present time, so it's not really, I'm not involved directly anymore, but I have noticed that the LMB, the goal as far as I can see is still to encourage people to tackle difficult and important problems and not be worried about short-term events like where is your next paper coming from. It's still very important to have a steady flux of talented students, postdocs, and group leaders, both coming into the lab and going out of the lab, perhaps to start bigger institutes later on. We still have relatively small groups and no teaching, although some people do teaching because they feel it's an advantage. No necessary grant writing, although you can if you wish, and not very much administration. So that's quite an important thing. We still have mainly the MRC budget as a central budget. So groups do not have individual budgets. So if 2 students, for example, have a bright idea and want to do a new experiment, they don't need to worry whose grant is coming out of. They can just do it. And still there's a focus, of course, on retaining good people, on mobility. We've got the canteen, we've got the seminars, and then another idea which is probably illustrated by the spin out in genome sequencing. Once something gets too big and monolithic and requires lots of resources, the idea would be to spin it out into another institution or a company or a startup so that the lab can focus on the small scale research and constantly looking for new ideas. And the challenge is at the moment, there are quite a lot, and obviously Jan has got to address these. Many of the issues for LMB relate to how the LMB, in the changing climate of science, how should it relate to the outside world, local people in Cambridge, or to the UK, or to Europe, or the world. And obviously now there's a lot more money going into science, so there are a lot of international initiatives where vast resources are put into a particular activity. And so it's very important that individual labs should develop collaborations with those. For example, genome sequencing, Drosophila, Dictyostelium genomes. So that's a sort of international climate. And then domestically, there are changes as well. So for example, recently, a year or so ago, the research councils in the UK have been put under one new umbrella organization called UKRI, Research and Innovation, and Medical Research Council is still in there, but it's not clear how that new umbrella organization and how that will develop with time. That's still under discussion. It's an important factor for looking into the future. And then we have Brexit. So UK is still part of Europe, but not in the EU. Doesn't affect many things because many organizations are not part of the EU. So EMBO, EMBL, ESRF, ILL, and these are organizations with their own independent existence. But there are others like the European Research Council, which is genuinely part of the EU, and relationships between the UK and the say ERC need to be worked out. So Jan has many challenge, and perhaps in a future Sister Labs talk, we will hear a little bit more about that. So I'd like to just finish that. So hopefully that's given you a little bit about the history, how the lab started and how things were run in the olden days, how they were maybe 20 years, 30 years ago, and a little bit about what the problems are and the issues now. So I thought I'd just finish by showing you the 3 stages in the evolution of the lab by showing you a photograph of the buildings that people were in. So the first one is the hut in the physics department downtown in Cambridge. It was built for another purpose, low temperature physics, never occupied by them, but Max persuaded Cambridge University that it should be used to house the fledgling MRC unit, eventually Laboratory of Molecular Biology, probably 20, 25 people in it. It was just a hut with a tin roof and some cars. And then in 1962, they persuaded the MRC to build this new building. This is on Greenfield site, where now Addenbrooke's Hospital, which covers all of Cambridgeshire and East Anglia, there's probably 100 people in those days in that building, but now there are 25,000 people on the site. You can see there's one bus stop with one bus and so it's all now a big, big site actually. But that was again helping to open up a new campus on the southern side of Cambridge. And then finally, we have the building that we're very lucky to be in now, largely funded out of income from commercial income from monoclonal antibodies. It's on the about 800 meters west of where the old lab is. The railway is in the background, and we're part of this new campus. And then I'm just going to finish with one thing. You can see the chimneys for the ducts and the fume hoods and where the air comes in. But if we look at one of them in closer up, you can see that there's a bird of prey, probably a sparrowhawk, decided to nest there last year. So even the wildlife likes our new lab. So hopefully that's given you a bit of an overview of the lab. Thank you very much.
Thanks so much, Richard. I think that was a fantastic look into the evolution of the LMB culture. We do have a few questions. I want to start with, can you tell us a little bit about what the opportunities are as far as student programs at LMB, whether that's undergraduate, graduate programs, internships?
Yes, it's, I think, very professionally run now. So when I was a student, in 1966, I wrote to Max Burutz, and you know, it took a long time to reply. It was very informal. And he said, oh, why don't you come and visit? I said, okay, why don't I come next week? He said, no, it'd be better if you came Saturday in 2 weeks. And it turned out that was the mechanism for recruiting students in 1966. They had an open day, 2 hours on a Saturday morning. And then 2 students were recruited that year. Now there are about 30 students. There's a complete timeline. You can look on the lab website and find out how to do it. But we have working visits for school children. We have summer studentships. We have undergraduate university, people can do it part of the degree. And then there's a PhD program with this 20 or 30 people a year. So overall, there are probably 90 or 100 students. And it's all on the lab website and all very smoothly run as far as I can see.
Wonderful, thanks. You also mentioned the desire to increase your international collaborations. Do those generally happen? I mean, I know they happen organically, but is there some sort of programmatic facilitation of these international group collaborations, or is it just solely reliant upon, you know, the organic interactions between scientists in different countries?
Yeah, I think it's done on a one-to-one basis, so that, for example, if there was one of the ones I remember I mentioned, Rob Kay was a cell biology group leader working on dictyostelium, and then there was a worldwide dictyostelium genome effort, and they were all very well involved with that. Similarly with Drosophila, there's a strong LMB work in that, of course related, joined to Janelia, but there's a big worldwide community in that. Then there's a nematode community, a cryo-EM community. I would say they're all have an independent critical mass, momentum, and existence, and probably there isn't what you might call an MRC-led program to do that, although Jan may be able to fill us in on that better.
I'm going to ask just one last quick question here. Actually, I may let Ellie ask her question. Ellie, are you there?
Yes, I'm still here, but you couldn't hear me before.
Oh yeah, no, we can hear you now. Go ahead.
Okay. Yeah, my last question was, does the LMB collaborate a lot with Big Pharma? And if so, how does that kind of work?
No, I can't hear that, I'm afraid.
So Ellie's question, sorry Ellie, we lost you there. Does the LMB do a lot of collaborations with Big Pharma, and how does that work? How is that facilitated?
Yes, there has been, as I mentioned in this, there's a sort of change in, there's much more interaction now, both with companies that have been started by people in the LMB, and not everybody likes many people want to get on with their own work, and they'll patent something, and then that's the last they want to hear about it. Other people take more of a hands-on interest. So in the past, one of the company startups, we had ribotargets. John Carn took half time in 5 years to go and help get it started, and then came back into research. And then I think there is a desire on the part of a lot of big pharma companies now to develop. So they were relatively isolated, say 10 or 15 years ago, and it cost them a lot of money to do sort of innovative, cutting edge, blue skies research inside the companies. So I think many of them now prefer that the basic long range sort of gambling type of blue skies research is done in academic labs. And so they will often give support for 1 or 2 postdocs. And so there's a proportion of that inside the LMB. I would say, I mean, and Jan can correct me, but I would say it's probably 80% of the funding comes from the MRC. Then there's a significant number of groups that have Wellcome Trust or other charitable support. And then probably there's maybe, I don't know, 5 or 10%, a little bit of support from industry for particular projects that they're interested in. And then obviously people get recruited out of the lab into companies, either the LMB startups or into big pharma companies. So there's a lot more interchange and toing and froing between big pharma, little pharma, and the LMB than there ever was back in the 1960s. And so, for example, in Cambridge, one of the initiatives recently, because of the cryo-EM, that particularly Shores and you know, the programs, 5 pharmaceutical companies in Cambridge decided that they would like to create a pharmaceutical consortium to do cryo-EM, thinking about drug development in the long term. And so they bought into that, with LMB acting as a kind of midwife role to sort of introduce them to that technology. And it's been running for about 3 or 4 years now. It's been very successful. All the companies have bought their own equipment, but they've also expanded the consortium. So I would say, looking forward for the LMB and possibly for other labs, that it's very good to be light on your toes, fast on your feet, in terms of deciding how deeply to get involved with both big pharma and small pharma.
Great. Thanks, Richard. I think that's all the time that we have today, but I want to end by once again thanking very much the speakers, Michelle, Shores, Richard. The talks were fantastic. Thanks for joining us. I encourage everyone to go ahead and click the link in the Zoom webinar chat box and head over to our Slack channel, because the speakers have promised me that they will head over and answer any questions that you didn't get to ask today. And last but not least, I want to let everybody know that next week we will be highlighting EMBL with speakers Nassos Tipas and Johanna McIntyre. And thank you to all of the audience today. We had about 500 people on the call. And thanks everyone very much.